Neural crest cells exhibit different levels of multipotency. In the case of the trunk neural crest cells, experiments using the confetti mouse model, in which nascent trunk neural crest cells in the mouse were fluorescently labeled using transgenes, show that a single cell could give rise to different cell types that populate different tissues. So for example, as you can see, a single orange cell that's going to be labeled before the cells delaminate is eventually going to be found to give rise to descendants that populate the dorsal root, that become melanocytes, and that become Schwann cells of the ventral root. And according to the results of this experiment, it was found that most trunk neural crest cells, approximately 75% of them, appear to be multipotent, and a small population of them appear to be already unipotent when they leave the crest. So the conclusion of this experiment was that the mouse embryonic trunk neural crest cells appear to be multipotent stem cells. Based on several studies, it has been proposed that neural crest cells are originally multipotent, but their fate becomes gradually restricted as they encounter paracrine factors during their migration. Although neural crest cells become apparent during neurulation, their induction happens during early gastrulation. And this is happening at the region that is the border between the presumptive ectoderm and the presumptive neural plate. In the anterior portion of this region, the ectodermal placodes form, which give rise to other structures like the lens of the eye and the inner ear. In the case of the neural plate border that's going to give rise to the neural crest, it is thought that there is an interaction that's happening between the epidermal ectoderm, the neural ectoderm, and even the underlying mesoderm, for example, the notochord, that is responsible for inducing the neural crest cells. And this interaction among the prospective epidermis, the neural plate, and the underlying mesoderm involves paracrine factors, specifically Wnt, BMPs, and FGFs, that are going to activate transcription factors that will specify the neural crest and induce their migration. If you recall from our discussion on amphibian and mammalian development, if both Wnt and BMP signaling are continuous, the ectoderm is going to develop or differentiate into epidermis. But if BMP is inhibited by inhibitors like chording or nuggin, the cells will become neural cells. It has been shown that for the neural crest to be induced, Wnt signaling has to induce BMP, and Wnt signaling needs to remain on. And this in part is important for modulating the levels of BMP so that they are intermediate. And if you recall from our earlier lectures, intermediate levels of BMP are needed to specify the neural crest. So the specification of the neural crest then involves these paracrine factors, specifically Wnt and BMP, that are going to lead to the activation of transcription factors in that neural plate border region. These transcription factors activate a second set of transcription factors that are known as border specifier transcription factors. And the border specifier not only will activate or be necessary to give the border cells the ability to become neural crest cells, but also to give them the ability to become dorsal neural tube cells. So this is a more general set of transcription factors that will then activate a second set of more specific transcription factors, the neural crest specifier transcription factors. And these include FOXC3, SOX9, and SOX10. And if you were to express these neural crest specifier transcription factors in locations that were outside of the border, so in other regions of the ectoderm, you would form ectopic neural crest cells. And that suggests that these neural crest specifier transcription factors are required for the specification of the neural crest. Now, the neural crest specifiers are going to give the neural crest cells the ability to migrate, and they will also activate the transcription of other factors like MITF, for example, that are specific to a given lineage of neural crest cells. So for example, MITF is activated in those cells that are going to become melanocytes, whereas collagen 2A1 is going to be activated in those cells that become chondrocytes.